Well, good morning, everyone. Today I'm going to summarise the Trichoderma radiata pine research program that is based at the Bioprotection Research Centre at Lincoln University. Today I will concentrate on the contribution of Trichoderma to improving the growth of trees in New Zealand forestry. The Trichoderma radiata pine research program is looking to use beneficial Trichoderma root endophytes to enhance growth and suppress disease in forestry, nurseries and plantations. And if we are successful, this will lead to reduced chemical use and improved growth in nurseries. It will generate healthier forests with faster growth, and we may be able to get protection against diseases not here yet. The Trichoderma inoculants offer a non-chemical, a natural alternative for growth promotion and disease control. This is becoming increasingly important today and in the future, with the emphasis on minimising environmental impact and finding more sustainable ways to operate. First, I'd like to describe what Trichoderma root endophytes are. These are naturally occurring fungi that are found in the soil and in plant roots, and they have a symbiotic relationship with plants, so both the plant and the fungi are receiving benefits from each other. Now, hundreds of Trichoderma strains were harvested from the roots of very healthy plants, native cultivated plants from all around New Zealand. So not only from radiata pine trees, but also from other crops and ornamental and weed plants. They were tested for growth benefits and disease suppression in many greenhouse and plantation trials over a number of years. And this was the work that Dr. Robert Hill did. I'd like to point out that trichodermas do a similar job to mycorrhizal fungi that you'll be familiar with. So trichoderma have been shown to promote plant growth in numerous research or crop trials. It can improve nutrient uptake. It can increase the plant's tolerance to stress like drought. Trichoderma can also activate the plant's defense system and can be aggressive against pathogens. Now, the main difference between Trichoderma and mycorrhiza is that the mycorrhiza are generally seen on the outside of the roots, as you can see in the bottom slides. However, Trichoderma is generally inside the roots and is not visible to the eye. So if you look at the top middle slide, I have an image of Trichoderma hyphae inside a piece of radiata root, and it has been stained with a fluorescent yellow-green dye. Now, luckily, Trichoderma is easy to propagate and grow in the nursery, and I can now get very high yields of spores with my methods. Trichoderma is easy to use in the nursery, with the most efficient application method being as part of a seed coat, although you can apply it as a soil drench with conventional sprayers to the soil, to soil beds or to trays. Recently, I have been getting increased interest from the forestry nurseries to use these trichodermas, so this has been satisfying. Next, I will look at the impact of Trichoderma on tree growth in plantations. In winter of 2018, I set up 8 large validation trials, each being around 2 to 3 hectares in size. And the 2 most effective Trichoderma treatments were selected from a series of national trials. And these mixtures were named PR6 and PR3A, and they were compared to an untreated control. The trichoderma was applied as part of a seed coat in the nursery, and these seedlings, 1-year-old seedlings, were planted out in 4 regions: Gisborne, Northland, Nelson, Bay of Plenty, Waikato, and within each region there were 2 sites. The trials had a robust statistical design with large plots of 9 by 9 trees, and I have been sampling the middle 5 by 5 trees. And there was also a good level of replication with 7 to 10 reps per trial. Now, last year at the conference, I presented the data for each trial individually, and I showed that there were large, significant increases in height to trichoderma in all regions, apart from Nelson. So when this data was averaged over all trials, we found a significant 6 and 7% increase in height with trichoderma compared to the control. So this was a very promising start. I have just finished doing the 2-year measurements a few weeks ago, and I found a similar result to last year, with tree height being significantly increased by around 6% with trichoderma compared to the control. I also measured trunk diameter in 4 of the trials this year, and I found a larger significant increase to trichoderma of 9 and 10% compared to the controls. So we can conclude from these results that trichoderma mixtures, these 2 trichoderma mixtures applied as a seed coat in the nursery, are giving a significant boost to early growth in the plantation. I'm also starting to measure foliar disease in these trials, and this graph is a result of one of the trichoderma trials where the PR6 trichoderma mixture significantly reduced Dothostroma by about half. So trichoderma, it's not eliminating Dothostroma in this case, but it's keeping the disease at a much lower level. Now, there's been strong interest from the South Island forestry companies. So another validation trial has been established in Nelson this winter using their own stock, and 5 other validation trials will be established in Canterbury, Otago, and Southland this coming winter, in winter 2021. I have also looked at the impact of trichoderma on plant growth in nurseries. I have run 2 trials at the Tainai nursery in Rotorua using 2 important radiata clones that do not root well. And I found that the production values, the number of plants that could go out the gate, was significantly increased by between 45 and 75% compared to the controls, which were with and without fungicide. And this was mainly due to increased survival and root collar diameters at harvest, and also to improved root initiation and growth during the season. I also ran 2 trials at the Lincoln University nursery and found that survival and growth was significantly increased in Douglas fir seedlings, with up to a 10% higher survival rate at harvest and up to a 25% increase in height and a 42% increase in overall plant dry weight. There was also a trial at the Southern Cypresses nursery where I found that trichoderma mixtures resulted in up to a 27% increase in root dry weight in cypress seedlings. So overall, this suggests that trichoderma application can result in higher production values in the nursery, and these larger plants will lead to better establishment in the field. Now, other work that I'm doing is I'm working out whether trichoderma can control canker in cypress plants, and also looking at whether trichoderma can control Swiss needlecast in Douglas fir. I'm looking at the impact of nursery chemicals on applied Trichoderma strains and whether the chemicals reduce the colonisation of Trichoderma in the roots, because it may be that some nurseries wish to continue using some chemicals to reduce risk, so we need to know how sensitive Trichoderma is. Another important issue is, can Trichoderma be applied to established forests to increase growth and improve health? A large portion of the New Zealand forestry estate has had no Trichoderma, so is there a practical way of applying Trichoderma to these forests? Another project is, can Trichoderma be used to boost poor soils in nurseries before being brought into production? Just got a pilot trial has just started with this. And lastly, how well does the applied Trichoderma persist in the forest? And this is an ongoing project as well. So in conclusion, the two selected Trichoderma root endophyte mixtures have so far been validated with significantly improved plant growth and health in the nursery and plantation trees. Now, these trials will be monitored in the future. Early indications are that Trichoderma inoculated plantation trees may have less foliar disease than untreated trees. Again, with more monitoring work will be needed here. The selected Trichoderma mixtures appear suitable for the 3 forestry species that they've been tested with, and should be appropriate for other species as well. So this means that one mixture may be sufficient for the forestry industry, so this is a bonus. Nursery seed coating or drenching is the most efficient and effective way to introduce Trichoderma into the forestry system. And so overall, these Trichoderma inoculants appear to be an effective growth and biocontrol tool for the New Zealand forestry industry. And this has led to a business case being currently developed for one of these Trichoderma mixtures with a commercial partner. And lastly, a big thank you to the following companies. I'm highly reliant on help from these companies for access and to help establish and monitor the trials, so their help is greatly appreciated. Thank you.